Hi, I'm Jay Phelan. Let's talk about biology. I'm going to talk here about meiosis, the process by which sexually reproducing organisms create gametes, and how this process generates genetic variation within a population. So there are two ways that any cell can divide. We have asexual reproduction, where an organism can be single-celled or even multicellular, creates a new individual solely from the genetic material that is present in that individual. Or we have sexual reproduction, where two individuals are going to contribute, and we're going to see some pros and cons of each. So it's sort of a silly question, which is better, has to do with the conditions that a population finds themselves in. So for instance, you can look at a very important upside, a benefit of sexual reproduction, and that's that the process of producing the cells that are going to take part in it, the gametes, leads to a lot of variation from one gamete to the next to the next to the next. You've also got two individuals. So you have varied offspring that are being produced, and that benefits fitness as long as the environment is changing, so that you produce a lot of different offspring. Maybe one of them is going to hit it in this environment or be better adapted. But you've got some downsides. There are some really important ones. Only half of your genes are present in your offspring. You inherited something from your dad, something from your mom, for every single gene. And yet, when you create the offspring, you only get to put one or the other of those in. So only half of your genetic material is going there. It takes time and effort. You have to find a mate. You have to do the mating. There are opportunity costs. And this can require risky behaviors. In a lot of species, you've got to compete, you've got to do displays, you've got to maybe maintain a territory, all those different things. So you've got costs and benefits to sexual reproduction versus asexual reproduction. But the important thing is we're going to see that with sexual reproduction that there is a lot of variety among the offspring that are produced. In order to do this, we have to look at meiosis, which is how you produce the gametes, the cells that are going to take part in the process. So two important outcomes stand out when we look at meiosis. And the first is that an individual starts as diploid. So I've got two copies of every gene. I have two copies of every chromosome. I got one from my mother, one from my father. But when I produce gametes, sperm, if a woman produces eggs, they're only going to have one copy of everything. So there's a reduction in the amount. Secondly, every one of the gametes, every sperm, every egg is going to have some different combination of traits. So if you produce a lot of offspring, they're all going to be a little bit different. And these are very important outcomes, allowing sexually reproducing species to have a variety of offspring, and then in a changing environment, they are more likely to produce offspring that are well suited to that environment. So we'll look at the process step by step, and it's divided into two general phases, meiosis 1, meiosis 2. And in meiosis 1, what we're going to do is separate the homologs. I'll explain what that is. Both of the times we go through it, meiosis 1, meiosis 2, you can think of it as PMAT. I think of it like the MCAT, but it's PMAT. Prophase, metaphase, anaphase, telophase, and then we're going to do it again. So I will, for prophase, we have to break it down actually into early, middle, late. I'll explain why. So in early prophase 1, the chromosomes are going to start to become visible. This is because usually, if you're looking at a bunch of chromosomes, they are so uncoiled and diffuse, they look like a plate of pasta. And it's hard to see that they are linear, although you know that a piece of pasta is linear, a piece of spaghetti. So they'll start to coil up, and I like to think of this like taking your shoelace and you twist it, twist it, twist it, twist it, and it's still linear, but it gets shorter and shorter and shorter and thicker and thicker and thicker. That's why we can see it. In preparation for meiosis, we're going to see that suddenly the genetic material becomes visible. Now, the terminology that we're going to use here is a little odd in that it changes. So a chromosome is a linear piece of DNA that carries the genetic information. But we'll talk about sister chromatids because prior to the process of meiosis, every chromosome is going to make a duplicate of itself. Because think about it, we're going to be making additional cells. So we have to duplicate it. But when we duplicate it and they're stuck together, those two identical pieces of DNA are called sister chromatids. And then finally, we have homologous chromosomes. That is the version of chromosome number 1 or 2 or 3 or 4, whatever, all the way to 22 in humans, that I get from my mother is the maternal version. The version I get from my father, the paternal version. So they still have the same traits for each, but they don't have exactly the same features, my mother and my father. So homologous chromosomes have all the same genes at the same position or loci, but not necessarily the same alleles. Usually the chromosomes are going to be uncoiled, but as we approach cell division, they're going to coil up, become more dense, become more visible. And you see here in the diagram, after we have replication, they've duplicated themselves. They stay stuck together at the centromere. And so now this is why you see that familiar picture of chromosomes as an X, which is a little weird, right? Because a chromosome is a linear piece of DNA. So why does it look like an X? That's not linear. But every time you see it, it's actually sister chromatids. So they've duplicated themselves. It's a chromosome and a duplicated version of it. You look here, see? Bowl of pasta. Because they haven't coiled enough, condensed enough for us to see that they are linear, although in fact they are. We will refer to homologs. That is for any given chromosome number that has all the same genes at each position or locus. You have a maternal version and a paternal version. So they are called the homologs, or a homologous pair of chromosomes. After replication, that's the duplication of the genetic material, we call them sister chromatids, and they're joined by the centromere. So going back to our diagram, in early prophase 1, that is defined by the fact that they've coiled up and coiled up and coiled up. You know, you're twisting the shoelace until they become visible. In my diagram here, I have a blue chromosome and I have a green one. But we have 2 copies of each. This is a diploid cell, so I have one labeled with P for the paternal version. Let's say it's me, I got that from my dad, and one of the maternal version for the green one. And then for the blue one, I have the paternal version, the maternal. If this were accurate for humans, there would be 23 different colors with a paternal and maternal version, but it's too hard to draw, so I didn't do that. So we see them in early prophase 1, chromosomes become visible. I should say the sister chromatids become visible. Then in middle prophase 1, this is where a lot of action happens. You have the sister chromatids from the homologous pair coming together. Now that's a mouthful, because think about it. Sister chromatids. That's the duplicated, the chromosome and its duplicated identical version stuck together at the centromere. But then the homologous pair, so the maternal version and the paternal version. So now you put those on top of each other. That's called a tetrad. So there's 4 strands. You have the maternal and the paternal, but then each is duplicated. So in middle prophase 1, they come together in this tetrad, and that's when crossing over occurs. So you're going to swap some bits of the chromatids that came from your mother, some from your father. We can look at this. I've drawn them as an X. These are sister chromatids, and now the blue one is the maternal version, the red one is the paternal version, and they're going to come together. At each position on this DNA, they are going to have the same genes. They come together, and then they swap little bits so that the version of eye color that is on one of the chromatids that came from the mother can swap places with the paternal version. So now you've got this stretch of DNA that might have something, your mother's hair color or foot size, and now all of a sudden here is the paternal version of whatever, hair texture or something like that. So you end up with this stretch of DNA that instead of having exclusively things you got from your mother or from your father, it now is a combination. So they swap places. I've drawn them in here, and now if you look, you'll see that no one piece is going to be identical. This is what it looks like when you are looking through a microscope. You'll see that they are literally crossing over. The result of this is that you get 4 unique strands. If you were to look at just from the top to the middle to the bottom, it's some different combination of traits that came from the mother and from the father, a little bit of each. So every one of my gametes, sperm cells that I'm going to produce, is going to have this collection of genes from my mother or from my father. So late prophase 1 just means crossing over is done. Now we go to metaphase, so PMAT, PMAT. So metaphase 1 is when these tetrads come to the middle of the cell. We can remember the name metaphase, the metaphase plate. That's when you see them all lined up. And they line up there, but it's random whether, if you think of there being a top and a bottom, whether it's the maternal sister chromatids are on the top or the maternal. And you've got that for 23 different pairs, which is which. So it's all going to be kind of random. And that's important because one is going to be pulled to the top, one homolog is going to be pulled to the bottom. And that's important because in these two cells now, they're going to be getting different collections of alleles. So the tetrads, that they're called a tetrad because they're 4 strands, they line up at the middle of the plate of the cell, so we call it the metaphase plate. Here's what they'll look like. There's going to be a line there, and they're kind of compacted together. Prophase, metaphase, anaphase 1. So in anaphase 1, that just means that we start pulling them apart. And because meiosis 1 is about separate the homologs meaning homologous pair, the maternal and paternal, so we start to pull them one to the top, one to the bottom. Not the centromere splitting. This is just the maternal sister chromatids and the paternal. But I can't really call them that because crossing over occurred. But we pull one to the top, one to the bottom. Centromeres don't split. That's anaphase 1. You can see them here. They start being pulled apart. Spindle fibers are attaching. All sorts of physical processes are going on that pull them apart. But from a genetic variation perspective, we can ignore that. Eventually, they get pulled all the way to the opposite side. We see the cell membrane starts to pinch in. This is telophase 1, so P, mat, prophase, metaphase, anaphase, telophase. That's just characterized by it starting to split. And on each side, the top and the bottom, you see that since we've separated the homologs, now we still have 2 copies of every gene. Because, right, the sister chromatids, they are identical versions. There was replication occurred prior to us ever seeing, being able to see the cells. So then there is kind of an intermission period, interkinesis. Sometimes the genetic material will uncoil, sometimes not as much. Then we move into meiosis 2. This is where we're going to separate the sister chromatids because meiosis, it's from the Greek word, means to lessen. We are going from 2 copies of every allele to just 1 copy. So there's got to be a reduction division. So we first duplicated everything, and then we divided the cell in 2. So we still have the same amount. In the meiosis 2, we're going to separate the sister chromatids. So there is only 1 copy of every gene in the resulting cell. So prophase 2, genetic material coils up a bit, and the sister chromatids become visible again. Metaphase 2, they're going to move to the center, the metaphase plate, and now they're going to be pulled apart. And this time, the centromeres are going to be split. So in anaphase 2, you start to see them pulled apart, and 1 copy of each of the sister chromatids. And it's hard to call them sister chromatids at this point because during middle prophase 1 of meiosis, there was crossing over. And I define sister chromatids as these exact duplicated copies. So now they're not exactly that, but people tend to say the sister chromatids are separated and pulled apart, 1 to the top, 1 to the bottom. I have 2 here because we already split them once in the first division. So then in telophase 2, you pinch together the membrane. I've drawn tails on them as if we're building sperm here. There's other stuff that goes on to do that. I should say this, if you were producing eggs instead of sperm, there would be an unequal division of the cytoplasm. So the first time we did that division in meiosis 1, most of it would have gone to one and not the other. Then in the second, same thing. When you divide them, most of the cytoplasm would go to one and not the other. And so the 3 that end up getting not as much, they shrivel up and go away. But the 1 still makes it as an egg. So we don't produce as many eggs that way, but it has considerably more cytoplasm. I've just drawn in the male gametes here, put a tail on them. Critical thing here, you'll notice that each one now, there's the green chromosome, there's the blue one, but you only have 1 copy of each. So the result of meiosis is that we produce, they're called daughter cells. They have half as much genetic material. You only have 1 copy of every gene. They're haploid instead of diploid, that means. And each cell is genetically unique for a few reasons. Right? Remember, crossing over occurred, so no one strand is the same. That pulling one set to the top, one to the bottom is random. Which ones get pulled to the top, which ones get pulled to the bottom, and so on. because one of the biggest important features of sexual reproduction, and meiosis in particular, is that we're going to be able to produce varied offspring so that in a changing environment, some of your offspring are maybe adapted to that environment. We're interested in understanding, where does the variation come from? Why is it that our kids look like us, but not exactly like us? Why is it that siblings look like each other, but not exactly the same? So I call this obvious statement one, because it is obvious that people look a lot like their parents. Here's Tom Hanks, Colin Hanks. It's remarkable how similar they look. Or, we have here Reese Witherspoon and one of her daughters looking very, very similar to each other, but not exactly the same. Here we have Jeff Bridges and Lloyd Bridges, his dad, and then Beau Bridges. And they kind of look the same, but not exactly the same. So this is important. Two separate things that are going on there, that they look so similar because, this is genetics. They're inheriting alleles from their parent, but they're not inheriting the full set. And siblings are not inheriting the same set. That's why Jeff Bridges and Beau Bridges can look the same, and one of them is really good looking and the other is not as really good looking. It's weird how that can be. Here's Charlie, my son. He looks a little bit like me, but not a ton. And here's Jack, another of my sons, who looks a little bit more like me. And I'll show you something in a second, I think, that's interesting related to this, which is the second obvious statement, that siblings look a lot like each other. So they're getting their DNA, if they're full siblings, from their mother, from their father, from the same place. But because of the variation that is generated during meiosis, they're getting a different collection of alleles. So now you look at Jack and Charlie together and you're like, oh yeah, they do kind of look alike, but one of them looks like me, the other doesn't. So it's this weird mixture, and you want to be aware of where are the places where you could get more or less of the same alleles as your parent or as your sibling. Or we look at some of the Jackson brothers, and you see that they all look similar, but they don't look exactly the same. Or three of the very many Baldwin brothers, and they have lots of similar features, but not the same. This is really crucial that you have this variation. Where does it come from? Why isn't it exactly the same? Because that's an important thing. Jonas Brothers, same thing. They're clearly sharing some genetic material, but not everything. Or Serena and Venus, or two of Ernest Hemingway's granddaughters, Mariel and Margot Hemingway. So they look the same, the Kennedys. So we want to figure out, how is it that they're so similar, but they're not the same? And each one is different as well. So think about the process and what are the steps, the places where variation could be introduced. Because genetic variability within a population, so not within an individual, within the population, it's enhanced in several different ways through meiosis and sexual reproduction. At least 5 different ways, and let me give you one of them. My offspring aren't going to look exactly like me, right? Because somebody else takes part in the process. That's one, but think of another one. So when at anaphase 1, when we separate the homologs, there's a maternal for chromosome 1, maybe going to the top, but the paternal for 2 going to the top, and for 3, maybe the paternal again. And there's some, out of 23 pairs, some randomness of whether it's maternal or paternal going together. So that would be another one. When the homologs are being separated, which ones from the metaphase plate, maternal versus paternal, go to the top or bottom. But you should be able to think of at least 3 additional parts of the process where variation is introduced. So I'll stop here.